So low melatonin in autism was a replicated findings, first by this group who found this. So this is normal melatonin level, high during the night, low during the day, high during the night, and so on. And in the autistic group, there was low level of melatonin. It was replicated by a French group, and our group replicated the same aspect, that low melatonin in child with autism. And the parents were intermediate between the children and the controls. So to summarize, something like 3 years of work, and we have more data coming. We looked at the genes synthesizing, coding for the enzyme synthesizing melatonin, and we could find mutation in the ISMT. ISMT, is the last enzyme of the melatonin synthesis. And in his family, the father do not carry the mutation. The mother carries the mutation. It's also a mutation that truncates the protein. Normally it's a large protein, and it's now a short protein. And this is the normal here synthesis of melatonin during the night, and this is a synthesis during the night, for the mother and for the child. So two messages. The first one is that low melatonin is not a consequence of autism. I mean, we have now many families where there is a deficit already in one parent. But the second message is that melatonin does not cause autism. Melatonin does not cause autism. Melatonin does not cause autism, okay? So, because this mother, she has no melatonin, but she has no autism. And when we asked this lady, do you have sleep problems? She said, no, I don't have sleep problems. I can sleep 2 hours or 12 hours, I don't care. Which for me, I think it's a circadian problem. That she said that she goes to sleep when she's tired, but she has no clear circadian setting. So what we are looking at now is what is the correlation between the low melatonin and the clinical outcome. And what we found is that low melatonin was not correlated to IQ, to any of these social and verbal communication problem, but was quite, I mean, we have now 114 individuals, was quite correlated with stereotype behavior, which fits with the serotonin levels. So, once again, low melatonin does not cause autism, but still I'm interested in melatonin. And it's because the deficit is often observed in a patient with ISD, and I think sleep is a major concern for the parents. We have private mutations, CNVs and SNPs that are associated with this trait. And maybe there is an alteration of circadian rhythms or sleep. So the last part of my talk is trying to see what are the two aspects, the synaptic and the clock aspect of autism. So this is one hypothesis. So it comes from this paper by Tononi, who is trying to understand why sleep, which I think is a fantastic and wonderful questions. Why do we sleep? And one of the answer of Tononi is that he said that during the day you have your synapse, which, I mean, I hope that you will remember some of what I say. So your synaptic weight will be different from the beginning of my talk to the end of my talk. But then if I talk to you every day like this, your brain will be out, and you will have to downscale some of your synaptic weight. So sleep could be one way of downscaling the synaptic weight, and then to come back with a little memory of what you have done the day before. What I think in autism is that there is already a problem in synaptic potentiation. I mean, maybe too few or too more synapses, I don't know, because, I mean, it's very complex. What is inhibitory, excitatory, and all these things. But maybe also there is a problem in downscaling autism, in the synaptic weight. So I see autism as an overload, synaptic overload. And that could explain some part of the epilepsy that we're going to see in autism. So I see autism more as a hyper-memory syndrome or hypersynaptic overload syndrome. So what if we could downscale this synaptic weight? And that's what we can do using melatonin, for example. So this is the sleep pattern of patients with autism, and it's really remind of this free running pattern that you can see in patients with in a cave. When you're in a cave, no insight between of the light and darkness. And this is this free running. And when you give melatonin at 9, it's a 14 years old Japanese child with autism. It goes like this, but then you give melatonin at 11 p.m. and the child is sleeping during the night. So with Paul Greengrass in London, we launched the largest randomized controlled trial for melatonin in patients with neurodevelopmental disorders and impaired sleep. And we are doing the genetics, and I hope we will get some new results very soon. So why finding genes for autism spectrum disorder? My answer is to find the best environment. So let me introduce you to Theodosius Dobzhansky. It's a famous geneticist for saying that nothing in biology makes sense except in the light of evolution. He said that it's incorrect to think of an organism genotype as determining this phenotype. It is correct to think of the genotype as determining the reaction norm of the phenotype. So what is the reaction norm? The reaction norm is if I plot the severity of autism here and a change in the environment. If you have the genotype 1, whatever you do with the environment, the severity will stay the same. With genotype 2, if I change the environment, then the severity will be lower. So this famous equation, the variations in the phenotype of what you observed in the patient, equal the variation in the genotype plus variation in environment plus variation in genes environment. I think it's more like this in autism. So you may have some patients with the mTOR pathway. It's another pathway which could be linked to autism with microcephaly, for example. You can have the neuroligin orexin-Schenk pathway. You can have other pathways. And the idea is to find what are the pathways. For example, for the Fragile X, for the tuberous sclerosis patients, maybe the rapamycin is a good environment, pharmacological environment for these patients, but not for the Fragile X patients. The mGluR antagonists are maybe good for the Fragile X patients, but not for the mTOR patients. And melatonin is maybe good for melatonin deficient patients and not for the one who already have melatonin. I think early detection is a good environment, behavioral therapies are good environments, sleep at school are good environments. So I like genes, but I think environment is really important. And let me introduce one of my heroes, Adrian Bird. And I think it's really to tell you that things can be reversed to some extent. So what Adrian Bird has done, if you don't know this work, I mean, it's really amazing. That's why he's one of my heroes. I mean, it took this MECP2, the gene for Rett syndrome, and he introduced a mutation in the mice. And then, fortunately, he can do this in mice, and we can't do that in humans. But he can remove the mutation out of the mice. And so this is the mice here, which is very sick. I mean, she cannot move. And then the same mouse, then he removed the gene. And then he goes back to, he can reverse quite a lot. So when I've seen this movie, I was really surprised. And that's why I do science, to be surprised. So, and I think this is really helpful. Okay. And so one of my best environment, les amis, and first Marion Leboyer and Christopher Gilbert, and my psychiatric heroes. And so Marion, she's fighting in France to do some biology for psychiatric disorders, and including autism. And Christopher, my hero. Monica, Stanislas, brain imaging guys, Paul, the sleep guys, Frank, the language, Tobias, the guy who is working a lot on Schenk 2, Schenk 3, and is doing the mice. All these individuals in my lab, which are fantastic. And Roberto is a neuroanatomist who designed the SNP software, the one, I mean, he was doing this on holiday on the beach because he's a neuroanatomist, but he likes genetics. And Elodie, she was listening to baboons in Nigeria, and now she's listening to mice at the Pasteur Institute. And all these individuals are really fantastic. Merci beaucoup.